you guys for coming here. My name is Claudia Vacca. I work at the Hawkesbury Institute for the Environment, so HIE at Western Sydney University. And I work on Broccolini and the fungal pathogen that affects it, called Plasmodiophora brassicae. I am also very, very informal when it comes to talks, so please feel free to interrupt. So when we talk about broccolini, a lot of really important things that people don't know that it's actually a hybrid. It's not baby broccoli, but it is a hybrid between broccoli and gailan. It is a pretty high value crop in Australia, and it's actually the second most bought vegetable after bell pepper. I'm sure everybody's seen it in the supermarket. However, it is severely understudied when it comes to pathogen susceptibility, and it is susceptible to all common brassica diseases, but in Australia specifically, there's a really big problem with clubroot. So what is clubroot? Clubroot is a really huge problem. It's actually caused by a biotrophic protist called Plasmodiophora brassicae, which induces gall formation in the roots, which you can see over here. So the roots are very, very swollen. They act as a nutrient sink for the plant, so the plant becomes very, very stunted. And it can cause up to 100% yield loss, which makes our growers very, very unhappy. And the big problem is that once you have it in your field, it can persist in soil and water for up to 20 years. So once you have it, you can't get rid of it. So how do we tackle this problem? Well, there's some current management practices that have been used. One of them is liming. So basically making the soil more basic, because clubroot like acidic soil. The application of calcium and boron, and crop rotation. As of now, the only thing that has been proven to work is actually crop rotation, which in Australia is a problem for growers that grow broccolini because they are usually monocultures, so they only grow broccolini on those fields. So when I was asked to develop novel technologies, this is what I did throughout my PhD. I looked at soilless cultivation, so I looked at different substrates. I investigated the pathotypes that are present in our fields, so within Australia, and then I tried to dig deeper into the disease mechanisms, so looking at the metabolomics and the transcriptomics of the disease, so how it interacts with the plants on a more molecular level. I'm only going to talk about two aspects of this today. One of them is going to be soilless cultivation, and then transcriptomics later on. So for soilless cultivation, a lot of you may be familiar with this. It's basically just an alternative pathway to reduce disease. It involves growing stuff in glass houses, so where no soil is involved. In this case, they're using rock wool. It's a closed system, so it's easier to keep clean and controlled, and it also can be done anywhere. So if you have arable, like land that is not arable, you can just plant a glass house there. So at my university, I was actually using something a little bit more rudimental, which is this. It's a polytunnel. It's actually a much easier alternative to a high-tech glass house because it's a lot cheaper for growers to install one of these. So this is what I used. When I was looking at alternative substrates to soil, I concentrated on things that were commercially and easily available, especially when it comes to recycled materials. That was a big interest of mine, and renewables, because we want to create a circular economy and not waste anything. And then I had to look at some specific characteristics of these substrates, so drainage, nutrient retention, aeration, and I wanted things that had high pH. So when I set up my grow trial, I landed on these 13 different substrates, which are very, very different. We have common things like peat. And then there's recycled textile, which actually comes from the material that we find inside diapers. So I just tried to grow my plants on a variety of different things. My control was a standard pot soil plus cow manure because I knew that broccolini grew very well on that. And I just planted them for 4 weeks, and I grew them for 7 months. So this was a very, very long experiment. So when I harvested them, I looked at the dry weight to measure the biomass of the plants, and I immediately could see that in gray there's my control, and there were some substrate that were performing better when it comes to dry weight. One of them was biochar, the other one was mushroom waste, and then a mixture of sand and vermicompost. And in contrast, there were other ones that did not perform so well, like the recycled textile, which was actually I was actually looking forward to, but evidently was not good enough. Another thing that I really needed to look at was the broccolini production. Now, this becomes really hard and busy when you're comparing 13 different things. So this graph looks a mess. However, if we're looking at the start date of harvest, so the start of production, you can actually see that there's some substrates that started noticeably earlier compared to others. And actually, if we filter it, this is my control, and these are the earliest substrates. So there is a difference of almost 3 weeks between the start of production, even though the total yield per plant ends up being roughly the same. So this is actually good because it means that no matter where I grow them on, the plants will produce roughly the same amount of florets that we can harvest. However, there is some potential in some substrates that may have multiple harvests. So yeah, this is a more filtered out look at what I mean. So you can see the control in gray and then everything else that starts a lot earlier. Then I looked at nutrient analysis, and now this is very important because obviously we want substrate that allow nutrient retention within the plant and then give nutrient to the plant. These were all watered and fertilized at the same rate, so there was no difference in that, just what the composition actually was initially of the substrate. And you could kind of already see that some substrate like mushroom waste and sand vermicompost accumulate nutrients a little bit better, including calcium, magnesium, and boron, which are the ones that are important in combating clubroot because they are the ones that are used in current management. Now, I just needed to also look at the substrate versus disease because until this point, I just looked at the plant itself and its growth. I did not add disease yet. Plus, I needed to actually test rock wool because this is the most used substrate when it comes to soilless cultivation. And actually, Perfection Fresh, the company that owns Broccolini, sent me samples of their rock wool from their glass houses. So I not only had new rock wool, but I also had rock wool that was used for previous crop production and that was essentially being reused. Because as of now, used rock wool goes into the landfill. It cannot be reused. So I first added this experiment, and then I had an infestation of cabbage white butterfly that destroyed most of my plants. So I cried for a bit, or for about a week. I had to painstakingly take out all the caterpillars from the plants, and then I fed them to birds, but the damage was done, so I had to completely restart the experiment. However, a good thing that came out of this was that once I harvested, because I had already inoculated my plants, I could make a disease scale index for broccolini. So now I had an actual scale that I could use to rate my infection. So we repeated this and see who exactly would win. Whether my control, the mushroom waste, we have sand vermicosmos, we have biochar, 2 types of used rock wool, and a new rock wool. And then we have clubroot on the other side. So I did that, and then I looked at the biomass and disease. And we actually can see that the used rock wool compared to the control did a lot better. And it doesn't matter whether it's diseased or not. It just did a lot better. And this is when it comes to biomass, where we're comparing dry weight of the roots and the shoots. So the plants have been harvested, the soil has been washed off, they've been dried, and I measure the weight. Once I look at the production of florets, I actually had a lot of stuff to compare it to, not just substrate. I had disease as well, but I also had other variables, like I needed to test for crunchiness because that is important for industry purposes. Whether a floret is crunchy or not will determine whether that floret can be sold. And also how big the floret is in terms of height and diameter. So those were all things that I needed to take into account when looking at the total yield. And I had to take a look at what's actually influencing the yield and what is not. And everything that's on this side of the graph is stuff that is influencing the yield, and everything that's on this side of the graph is stuff that is, positive and negative influence. And the things in red are the things that are positively, that are significantly relevant. So crunchiness, actually, the more crunchy it is, or in this case, the less crunchy it is, the worse it is for yield. So obviously, like the floppier, the broccolini is, the less it will weigh. But also, obviously, the more diseased the plant is, the less it will yield. And surprisingly, vermicompost and biochar also also showed a really big negative impact on yield. While used were cool, I put tomato and cucumber because that's what they were used for previously. It seems like the tomato one does better, but it is not significantly, it does not do significantly better. However, my effect size, I didn't have a lot of plants for the control, so that unfortunately kind of, yeah. It affected my p-values quite a bit. The experiment just needs to be a lot bigger, but it showed some really promising results. So when we're looking at final conclusion, we can definitely say that used rock wool improves biomass, and it probably improves yields as well. And the most important thing is it does under clubroot infection too. Mushroom waste and vermicompost also enhance growth, but do not enhance yield. And most importantly, substrate choice is influenced by the nutrient profiles, especially when it comes to potassium, boron, and sodium. So these were the three nutrients that had the most effect on the plant, that were most affected by substrate choice. And yeah, then I did my multivariate modeling, which confirmed that only a few predictors had strong independent effects. So I think as a really big takeaway, we can say that recycled and waste-derived substrates have a real potential for sustainable broccolini production. So going forward into soilless cultivation of broccolini, looking at used materials is the way to go. Next, we're going to transcriptome, so we're digging deeper into the problem. And by that, I mean, I had to address these questions. So what are the infection responses of broccolini when they are compared to its parental lines? Is there a difference in the pre-symptomatic infection phases? And can we find resistance to inform breeding practices? So this is a very long-term goal because breeding practices to breed new varieties takes years, if not decades. However, what I wanted to focus on was within the life cycles of clubroot was to look at the early time points, so pre-symptoms, before you actually see the club, the clubs forming. So I set it a time course, and this is actually broccolini, broccoli, and galan, all at 1 day post inoculation, which is 4 weeks old. And they look quite different, and that's just because the plants grow at different rates. And this is what they look like at 34 days DPI. And you can kind of see a really tiny gall here. And you might ask, why is it so small where you had bigger galls in the other experiment? And that's because I grew these in pots that were about this big because I had to grow a lot of them, and we don't have much space. So this is what the other brassica varieties look like, and you can see that the structure of the root system is quite different. So this would probably affect the club root club formation as well. So I actually had to define the infection stages under microscope because you could not see them in the naked eye, like you can't really see them. And I had to do that with a compound because our confocal was broken, which is why the pictures look like this, unfortunately. But what you can definitely see is that we have three distinct phases. So one phase is when the spores adhere to the cortex of the root hair. There's an early secondary plasmodium phase that I could distinguish, where basically the pathogen has penetrated the root and it's colonizing the root, colonizing the cell within the root. And then there's a mature secondary plasmodium where it's mature, it has taken over most of a cell, and it's about to burst and release more spores. So I found, I defined these three pre-symptomatic stages, and those are the ones that I selected for RNA sequencing. So looking at the what genes are activated during these phases. So it's kind of like a snapshot of what's happening. An interesting thing, just looking at the microscope, is that Guylan had a much delayed progression of infection. So the adherence to the cortex or root hair happened a week later compared to Broccolini and broccoli. And so everything was shifted towards everything was shifted in time. And because I had a lot of time points, I could definitely confirm that on multiple samples. So this is probably exactly what's happening. So once I had this, I did a RNA-seq, and I looked at what are the genes that are just completely different between Broccolini, broccoli, Guylan, which genes do they have in common, and does the gene expression change in time? So you can actually see here that the early secondary plasmodium stage and the mature secondary plasmodium stage, we have a shift in gene. First, the Guylan has a very unique genetic profile, and then that decreases while the unique genes for broccoli and Guylan increase. Now, big question, there's no root cortex adherence stage here, nor there is a control, and that is a huge problem. And that happened because all my RNA degraded. So I am currently in the stages of just repeating this experiment because I'm missing the very first time point, which is incredibly frustrating. But it did give me a really good idea of what we're looking at just slightly later on. So we have an idea of what the number of unique genes we're looking at, but what are we actually looking at? We need to do a gene ontology study, so exactly look at what genes are there and what they've been, what ontology terms are associated with them. And most of them are in the transcriptomics factor category and protein kinase superfamilies, and these are all genes related to plant immune response. So it makes sense that the plant is recognizing that there is an infection in action and that it's mobilizing an immune response. We definitely need to look deeper into what these genes are, and that's what I did later on, although most of these are not defined. So we're gonna have to start defining them. And then I looked at what are the genes that are up and down regulated throughout. So this is a very busy graph, as usual the genes regulation maps are, but I'm gonna break it down. So when we look at the early time points, in Broccolini there's a strong upregulation of genes, which is shown, the upregulation is shown in red and downregulation is shown in blue. So there is a shared area where all genes, all those genes in common are downregulated. And then there's an area where there's a difference. So you have Guylan genes downregulated with Broccolini and broccoli upregulated. And for mature, you kind of see a really, really strong cluster of genes that are highly upregulated, especially when we're comparing it to the earlier stages. So there is like a strong transcriptional response here, probably because of as the infection progresses. Well, Broccolini shows a milder pattern, and Guylan an even milder one. And in Guylan, the mature stage is just mostly neutral or downregulated, and there are even some signals that are like completely, and in broccoli there are some signals that are just completely absent, like they're almost at zero. So there are definitely like some differences here. Like the important thing is to look at what genes we're talking about, because this is just a map. But it's a lot of work going gene by gene, because we're talking thousands here. So I did a GoTerm enrichment specifically of early versus mature secondary Plasmodium, and as you can see, like most of them are unannotated. So we just don't know what most of these genes do, because there's no annotation on any of the databases that I checked. But most of them are transcription factors. So there is definitely a transcriptional response, and there's a very interesting pentatricopeptide repeat-containing protein, or at least 8 of them, in Guylan. And these proteins usually play roles in RNA processing under stress, so it's still kind of an epigenetic response that is downregulated. I couldn't just look at the plant side, I needed to look at the pathogen side too. So this is the pathogen side. For the pathogen side, I actually had cortex adhesion for Guylan. And yes, the pathogen is producing something really strong there that is just not present at all in Broccolini, broccoli, in any of the later stages. So these are probably effectors that that clubber is producing within the plant, and I really want to know what this is, and for that I need my earlier time points. So like because the Broccolini broccoli have an increased pathogen expression profile, it could indicate that there's maybe a delayed recognition or a breakdown in containment, perhaps, because this is like there's a heavier response later on, where Guylan has a very strong response early on, and the infection is delayed. So what is probably happening is Guylan has an early response, the infection cannot go on. Broccolini and broccoli ignore it, so the infection does go on, and then you see a delayed response in Broccolini and broccoli, while Guylan eventually just cannot probably mount a defense that is so efficacious, because clubber is a very, very vicious pathogen. So yeah, these are all like the areas that I have highlighted, and they just all produce differences. And Guylan just seems to be upregulated differently compared to Broccolini and broccoli. So those key takeaways, again, are in regarding to infection timing. So Guylan has a delayed pathogen development. Those early infection stages are genotype specific. And then later stages kind of show gene expression convergence. So everything kind of evens out as the infection progresses, either because Guylan cannot withstand the infection, or because Broccolini and broccoli just catch up on the defense. Broccoli and broccoli, yes, they express more unique genes during mature infection. There is a pretty strong spike for pathogen genes in the cortex adhesion phase. And everything is kind of confirmed by the GoTerms, because all of them show enrichment in stress and regulatory processes. So this has some breeding implications that are not immediate, but the important thing is that we identify expression hubs and gene families, and so that we can actually inform broccolini improvement. So the next step is actually compared with a non-infected control and my earliest time point to integrate the metabolomics data, so we can integrate, we can actually see what the physiological response of the plant is on a protein and metabolite profile, and then go do the GO-term enrichment by pathway so that I can look at regulatory networks, and so like how these genes fit within the regulatory networks of the plant, and hopefully validate candidate genes through qPCR across the genotypes and infection stages, although I do think that's going to be beyond the scope of my project. But yeah, I just want to thank everybody in my lab. Dr. Donovan Coles, without him, I wouldn't have been able to even grow Broccolini. And then my supervisors and all the people that worked and helped me with code and just moral support. Future Food System Perfection Fresh for their amazing help on financial end.